I would like to thank the organizers of the 11th Annual World Congress DNA Day 2023 for inviting me to give a speech, Non-Local DNA Wave Optics for the Remote Bioholography. I am a president and founder of the United States company Wave Genome LLC, incorporated in the state of Connecticut in 2010. I had created the fundamental theory of DNA wave optics in 1985, long prior to anybody in the field of bioholography, based upon my unified theory of wave crystals. DNA wave optics theory provides the theoretical basis for the remote management of biosystems via remote bioholography. Wave Genome LLC develops technology for the remote bioholography. Wave Genomes LLC bioholography technology is based upon the refraction code of wave optics in the scalar wave diffraction grating. Wave Genome LLC produces and distributes products and services of the remote bioholography. Wave Genome LLC offers the remote bioholography at close and long range. Products of the remote bioholography include digital and analog products as generators of the form and their combinations. Wave Genome LLC had pioneered the holographic digital pharmacy. Holographic digital pharmacy consists of A. Digital bioholograms of client recorded and transmitted by optical and non-optical lasers. B. Digital bioholograms of healthy physiological systems, of stem cells, etc. And C. Digital bioholograms of pharmaceuticals and supplements. Wave Genome LLC had developed various devices for the distribution of the holographic digital pharmacy. For the distribution of holographic digital pharmacy, Wave Genome LLC offers first a large-size medbed, such as the Ra sarcophagus. Wave Genome LLC was the first company in the world to pioneer large-size medbed based upon wave optics. In 2010, when Wave Genome LLC had pioneered its large medbed, it was the only company in the world offering a large medbed. Also, for the distribution of the holographic pharmacy, Wave Genome LLC offers second the Ra helmet. The Ra helmet offers the opportunity to use the DNA wave optics in a portable format. For the distribution of the holographic pharmacy, Wave Genome LLC also offers third the electrode-based chip structured on nano-level via laser by client's digitized hologram, modulated by various modulators, including stem cells. Electrode-based chips function as small-sized lenses to correct client's DNA wave optics. Wave Genome LLC was the first company in the world to A. Code electrodes with client's unique wave matrix, B. To process this wave matrix as biohologram, and C. To record and transmit this biohologram upon the electrode via laser. Also, for the remote distribution of the holographic pharmacy at a long range, Wave Genome LLC offers HoloNet, holographic bio-internet. I was the first scientist in the world who have succeeded in proving by clinical trials that an electrode-based chip encoded by the digitized hologram of the pharmaceutical drug on nano-level by laser and the digital bioholographic pharmacy function better than the conventional pharmaceutical drug. In October-December 2017, I have conducted clinical trials for two chips of electrode nature structured on nano-level via laser with the digitized hologram of aspirin and the digitized biohologram of the human embryonic stem cell. Scientific group headed by me consisted of scientists from the Moscow-Bauman State Science University, the largest and most important science university in Russia on the MIT level. We used the newly invented Russian laser spectrograph. The results demonstrated that Wave Genome LLC has created the effective digital anticoagulant that has the effect of aspirin without the side effects and danger of overdose. All pharmaceutical anticoagulants increase the time of coagulation. The clinical trials of aspirin, holographic aspirin, had demonstrated the increased time of coagulation by the digital anticoagulant for the first time in the history of world science. Let me explicate my fundamental theory of the DNA wave optics. The world is energy for 93% and particles for only 7%. DNA itself operates via the remote bioholography in accordance with wave optics. Precisely because of this, Wave Genome LLC develops the wave optics-based remote bioholography. Between cell division cycles, chromatin exists in non-coherent state, but during cell division, chromatin gets literally crystallized, forming two chromatids, forming a chromosome. The structural coherence of the chromosome determines its functionality, while the structural damage in chromosomes is the major cause for diseases. The structural coherence of the chromosome has specific nature. The functional structure of the chromosome is a scalar wave. The structural damage in chromosome prevents it from creating a scalar wave. The chromosome consists of two chromatids combined together in the X form. The two chromatids intersect in the center where they exchange their genetic material. The same gene gets expressed in the functional individuals via Metacentric Chromosome and in dysfunctional individuals via Acrocentric Chromosome. Metacentric Chromosome are the X-shaped chromosome with two arms that are equal in length. Acrocentric Chromosome are chromosomes with two arms that are not equal in length, meaning they exhibit gross asymmetry. The same gene in apes is expressed by the acrocentric chromosome and in humans by a metacentric chromosome. Metacentric chromosome is analogous to a well-centered and focused eye, while acrocentric chromosome is analogous to a myopic or far-sighted eye, out of focus. Ability of chromatids to exchange information and to regenerate and replicate in general depends upon the geometrical wave optics of a chromosome. If this geometrical wave optics of a chromosome is violated, the chromosome loses its ability to regenerate and replicate. The same gene is expressed by the acrocentric chromosome in the wild horses and by the metacentric chromosome in the domesticated horses, which are superior to the wild horses. Children with Down syndrome differ from normal children by the presence of extra acrocentric chromosome. Only because female gender chromosome is metacentric, a woman is capable of replicating, giving birth, and only because male gender chromosome is acrocentric, a man is incapable of replicating, giving birth. So the male chromosome, Y chromosome, is acrocentric, degenerative, and dysfunctional, and X female chromosome is metacentric, regenerative, and functional. Humans are water for 85%, but because universe is energy for 93% and particles for only 7%, our chromosomes are also the liquid crystal media for 7% and the wave crystal media for 93%. In order to restore the structural coherence of chromosomes' liquid crystal media, we need first to restore the structural coherence in chromosomes' wave crystal media. Each biohologram is a non-local lens with the specific wave optics in its wave crystal media, aka wave crystal. Instead of the vague term of quantum entanglement, I offer the precise term of refraction entanglement. Remote bioholography is based upon the refraction entanglement between two bioholograms. Refraction entanglement occurs between two bioholograms as non-local lenses when they have identical wave optics in their wave crystal media, aka wave crystal. Wave crystal media, simply wave crystal, is a state of reproduction and regeneration. Wave crystal is also the matrix point. In the state of reproduction and regeneration, the internal media achieves its most coherent state described as spatial translational symmetry. Spatial translational symmetry is a scalar wave. Scalar wave annuls all linear external signals when the peak of the forward-going wave is annulled by the trough of the same wave when this wave is reflected back upon itself. Scalar wave naturally merges due to polarization with the change of spin in energy torsion, so that every wave is always reflected back upon itself. Scalar wave emerges when the triggering signal is coherent, has its frequency stable, and is correspondent with its wavelength to the size of the resonator. So in the state of reproduction and regeneration, all linear external signals are annulled by the scalar wave. Thus, nullification of any external signal can be defined only as zero signal or zero energy. If the trigger signal is consistent, there emerges the scalar wave diffraction grating. The emerging scalar wave diffraction grating gets scaled. Every segment of the scalar wave becomes itself a scalar wave. Scaling of the scalar wave diffraction grating goes instantaneously ad infinitum to macro and micro dimensions. Every segment of the scalar wave is a resonator of a certain size corresponding to a specific wavelength. Since the scaling of the scalar wave diffraction grating goes ad infinitum, only the scalar wave diffraction grating can annul simultaneously the extremely high frequencies and the extremely low frequencies. The simultaneous coexistence and synchronization of all the segments of the scalar wave diffraction grating cannot be described only by one frequency. The simultaneous coexistence and synchronization of all the segments in the scalar wave diffraction grating can be described only by refraction. Scaling of the scalar wave diffraction grating is refraction with certain wave optics. Refraction is towards the one and only focus of the wave crystal zero center of the quantum nonlocality, one and the same for chromosome and galaxy. Thus, in the state of reproduction and regeneration, there emerges zero energy, which is non-local and instantaneous in its nature. And zero energy cannot be described in terms of electromagnetism, vibration, waves, and resonance. Zero energy is defined by the holographic principle, which states that the universe is entirely in its every matrix point. The holographic signal has the infinite number of waves, particles, and fields simultaneously. This explains the nonlocality of the scalar wave diffraction grating. The holographic signal is the zero signal. The zero signal is shared instantaneously and remotely through the focus of the wave crystal, zero center, identical for chromosome and galaxy. The zero signal is shared simultaneously to every matrix point in every dimension, ad infinitum in time and space. In 2012, I had formulated the implication of the holographic principle. If the universe is entirely in its every matrix point, then every matrix point is not simply different from any other matrix point, but is unique. So every matrix point biohologram is both non-local and unique. Matrix points differ from each other by their refraction codes. And if there are two copies of the same non-local and unique wave matrix, the information is transmitted instantaneously and remotely between these two copies, since they constitute one and the same systematic whole in the quantum nonlocality. So my theory rejects the purely biochemical interpretation of DNA, which does not account for the fact that the world is 93% energy and particles for only 7%. The biochemical interpretation of DNA cannot explain the remote and instantaneous transmission of the morphogenetic information by DNA, as described in the experiments by Dr. Alexander Gurevich, Dr. Cleve Backster, Dr. Vlail Kaznacheyev, Dr. Glen Rein, Professor Si-Chen Lee, Dr. Luc Montagnier, Dr. Peter Garyaev, and Dr. Konstantin Meyl. My theory also rejects the purely electrochemical and electromagnetic interpretations of DNA, attempting to describe DNA in terms of some resonance. My theory rejects The electromagnetic interpretation of DNA is failing to explain the holographic principle, which states that the universe is entirely in its every matrix point, meaning that the holographic signal has inside itself the infinite number of particles, waves, and fields. My theory rejects the hypothesis of DNA as a radio wave working on the principle of resonance between molecular oscillators, as not accounting for the destructive wave and interference, and so incapable of explaining the instantaneous synchronization, even in the case of the anti-phase cancelling of a signal. And that is why my theory also rejects the hypothesis of the molecular oscillators as simply the vortices, since DNA is a scalar wave annuls any linear signal or vortex, and so cannot be described as the radio antenna. My theory rejects the interpretation of a DNA scalar wave as a magnetic wave, as incapable of grasping the quantum physics notion of zero energy and zero field, which are the simultaneous coexistence of the infinite number of particles, waves, and fields, in the instantaneous sharing of the infinite scope of information, while the focus of the non-local and unique wave matrix point as a tetrapole. Tetrapole has static electricity and static magnetism that are not shared via waves, but via the zero signal. I repeat, the zero signal is not shared via space and time, but via the quantum nonlocality or the zero field. Zero signal is shared through the zero point, which is the focus of the wave crystal. Again, the wave crystal is non-local in time and space. Success in reproduction and regeneration lies in the mastery over wave crystals.